Aging is a normal part of life. Even your cells age, but they're oftentimes replaced. Now imagine that there's a human aging process that destroys your memory. It leaves you confused, disoriented, unable to perform daily activities, and may lead to a complete loss of speech. Now this isn't normal aging. This is Alzheimer's disease. Alzheimer's disease, or AD, is a progressive degenerative neurological disease whereby neurons in specific areas of the brain, so the hippocampus and the cerebral cortex, are dying at a fast rate. Because of this, the symptoms of Alzheimer's progress worse and worse over time as the disease progresses. So Alzheimer's disease, or AD, is the most common form of dementia, and over 700,000 Canadians suffer from this disease, and this number is expected to rise in the future. But there is no treatment available for patients. There is no cure. AD is characterized by the formation of amyloid plaques and neurofibrillary tangles in the brain. Now this can be explained by a few mechanisms, one being an unstable mitochondria. So mitochondrial dysfunction affects the way that an electron is passed along the electron transport chain during cellular respiration. If processed incorrectly, this leads to an increased production of reactive oxygen species and can cause damage like what's seen in patients with AD. Previous research conducted in our lab has been focusing on a method to halt this progression of the disease, particularly using an antioxidant called coenzyme Q10, or CoQ10. So a water-soluble formulation of CoQ10 has been found to decrease the production of reactive oxygen species in a cell line. Also, it's shown neuroprotection in animal models with Parkinson's disease. An important point to note here is that we're using a water-soluble formula rather than an oil-soluble formula, which doesn't cross the blood-brain barrier. This means that it can't provide neuroprotection to the brain. This leads me to the goal of my current research. We're using an analog of water-soluble CoQ10, this is called UWindsor Q10, and we're supplementing it to a transgenic mouse model of Alzheimer's disease. And this is going to help us examine whether or not UWindsor Q10 is effective in providing neuroprotection for treated groups versus untreated groups. These results will hopefully further the understanding of the underlying mechanism of the disease and may even potentially translate to a treatment for human patients. Now, aging is inevitable, but being able to hold on to the memories that you've created during your lifetime is what Alzheimer's research is working to preserve.